So we're back in module 2. We've done the first two, which are the very first one was a review. The second one was mainly about water. We either gonna do or have done a whole lab about water, 2.1. And so we've dealt with that. We're gonna deal with pH, I think is the second one in this module. So maybe haven't done that one yet. Okay. So one of the ones that you're going to do at home, you're going to do a lab at home, which deals with enzymes, and we're going to try to talk about that when we get to enzymes. This is going to be long because this is carbon compounds. These are the things we're going to deal with throughout biology. Okay, I'm not saying you can and water also, but these are the big ones that are real important. Okay, so carbon compounds, they're organic. Okay, so when we talk about organic molecules, we're talking about molecules found in living things that contain carbon rings or long chains attached to a whole bunch of other atoms like hydrogen, oxygen, nitrogen, sulfur. Oh, wow, we saw those, didn't we? Didn't we say commit some of those to memory? Right there. So we talked about that one, that one, that one. Yep, we mentioned those, didn't we? Okay, so those are some things you need to kind of keep in your mind. Check out your infographic on that page. It talks a lot about the carbon compounds. We talk about biosynthesis. We are talking about the process by which living organisms produce large molecules from smaller ones. Okay. Now, what's cool is that when we make proteins from the little things that make up proteins, we put them together the same way that we put um, sugars together, the same way we put DNA together, the same way we put lipids together. Okay, now they're different things coming together, but how they go together is the same process. And we'll talk about that here in just a second. Okay, it's the same process. So, one of the things that's nice about organic chemistry, carbon chemistry, it's the same, no matter if we're dealing with carbohydrates, lipids, proteins, or nucleic acids. Okay, so here's what we're talking about with these chains. These are these big chains of carbon. This is a ring of carbon. It's understood there's a carbon. There's a carbon. Okay, whoops, don't go too far yet. Okay, so this is a I don't know if I can draw a carbon. There we go. Anyway, that's a carbon. There's a carbon there. There's a carbon there. There's a carbon there. So this is a carbon. Okay, every every place is a carbon. Okay, so if we were counting the carbons, and I should have been counting the carbons. So it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 carbons. So that's big. 15, I named that one. So 15. So this is C15H3O. Okay. Here's nicotine. You see what nicotine looks like. Here's ibuprofen. There's vanilla. Here's Tylenol. There's aspirin. Okay. So again, all of those are considered, each section to be where carbon's at. Pretty big. Organic molecules, compounds, they don't all have to be living. Like nail polish is not living. That's a butyl acetate. Some of the nitrocellulose is not living, but it's more biodegradable. Candle wax, you can think of beeswax. All your gasoline are hydrocarbons. All your soaps, some kind of fatty acid, something, or some kind of hydrocarbon. Most all your fabric is an organic compound. Carbon, our nylons, all organic. Okay, paper. All right. So here is kind of what that infographic was looking at. So there are carbohydrates, nucleic acids, proteins, and lipids that we're going to deal with. We're going to talk about a monomer and a polymer here in just a minute, and then this is the structure of where they're at. Okay, so sugars are carbohydrates. Here's a monosaccharide. Starch is a polymer. The granules in the chloroplast, and we'll get there. We're going to talk about photosynthesis. We're going to talk about carbohydrate digestion. So nucleic acids, a nucleotide. DNA is a polymer, chromosome structure. Protein is amino acids, polypeptide, and then like muscle fibers, all of that. Fatty acids, triglycerides, and then adipose tissue. Okay. Don't commit any of this to memory. We're going to do it here in just a minute. Okay, so that gives you an idea, picture of the 4 basic ones we're going to talk about. 3 of them we're going to really get in depth, and then nucleic acids we're going to talk about, but we're not going to get in depth with them until we get into genetics. Okay? All right, so basic terminology. Every one of these is made up of a monomer. So a monomer is the smallest molecule. Okay? So we put the monomers together, one unit of whatever this is, an amino acid, a simple sugar, a nucleotide, a fatty acid. This is one monomer of that makes a polymer. Okay, so all of them are made up of monomers. You put your monomers together, you make a polymer. Okay, the other term you need to kind of think about is macromolecules. This is what we're talking about. Macro means big. These are huge molecules. So poly means many, macro means big. Now, this does not mean that we can see starch without a microscope. All right, now we can see, technically we can see starch because potato starch, when it's broken up, is the starch, cornstarch that you use to thicken stuff. So yeah, so we're talking about big molecules. We can see them because they're put together with these little small ones. Okay, and we'll talk about that as we go through here, as we break them down and put them together. So an overall, and this is what I mean, this is the same reaction. So remember back in catabolism and anabolism, okay, way back in module 1, catabolism, cutting them up, anabolism, putting together, okay, back on page 18 in your book. So there's 2 processes. You got to put them together or you got to break them apart. The process of dehydration synthesis puts them together. Now think about the word dehydration. If you're dehydrated, you've lost water. So what happens is the water is going to be formed out of this. So you're going to link them together here by an oxygen molecule, hooking them together, and the water is going to come off. Okay, so it's dehydration synthesis. Okay, the other process this is anabolism. Okay, so that's anabolism. The other one, catabolism, is called hydrolysis. So hydro means, I hear it, water. And lyse, L-Y-S-I-S, not L-I-C-E, L-Y-S-I-S means to split. Okay, so it's using, we take a polymer and we use water and we split it down into its monomers. Now, sounds easy, but all of these are mediated by an enzyme, and we'll get there because we haven't talked about proteins yet. We're just giving you the basics. This is how amino acids are joined together. This is how 3 fatty acids and a glycerol is joined together for lipids. This is how glucose and galactose is put together to make lactose. It's always the same stuff. This is how 2 amino acids go together to get a protein. Always, always, always the same. You put them together, you lose water. Dehydration synthesis. You split them up, you got to have water. Okay, so that's why water's so important. Okay, so again, we're going back over it again because the processes work the same way all the time. So dehydration, water's a byproduct. Okay, refers to the formation of large molecules, polymers, macromolecules. Okay, the other term used to be condensation reaction, but condensation reaction can also mean there are other molecules that are coming together and are not just water molecules. Okay, so we're not going to get too technical, but phosphorylation, when we put ATP together, this is what we're talking about. And nylon are examples. Condensation refers better to them. We're going to use dehydration synthesis. Okay. So here it is. There's the water coming out. There's the 2 molecules together. Here's the water. Okay. So hydrolysis, putting the water in, splitting it. Remember, hydro means water. Lyse means to split, to separate. You got them back again. Okay. And in some reactions in our body, this is constantly going back and forth, back and forth, back and forth. And we're going to talk about this more in depth. We got to know dehydration synthesis, hydrolysis. Dehydration synthesis means to make. Lyse means to split. Just kind of keep that in your vernacular. Quoting Chaucer here, in our vernacular, one of our words we had to learn in literature. Keep that there. So the first ones we want to talk about are carbohydrates. Okay, so carbohydrates or organic molecules that contain carbon, hydrogen, and oxygen. It says it right there. Carbon, hydrogen, 8 means oxygen. Okay, so hydrate means to add carbon, carbohydrate to add water. So monosaccharides, disaccharides, polysaccharides are the ones that we're going to be talking about with carbohydrates. So they come in 3 distinctions. Monosaccharides, of course, are the mono units, the monomer unit, and then polysaccharides are the big ones. Now, their function, carbohydrates' big function is energy. Okay, energy storage, energy release. Sometimes carbohydrates are structural components in cells, such as cellulose, chitin, and those things. So carbohydrates are what we're talking about. We're going to get specific, so get ready. Monosaccharides are simple sugars: glucose, fructose, galactose. These are called hexoses, meaning they got 5. Okay? No, 1, 2, 3. Yeah, they got 5. 5. All right. So they're the monomer unit of more complex carbohydrates. Okay. Or in the case of deoxyribose and ribose, these are pentoses. Hexoses is 6. Pentoses is 5. There we go. Now I got it. I was looking at fructose. Anyway, pentoses are houses. But actually fructose has got 6 because there's a 6th one right there. Okay? So hexoses, 1, 2, 3, 4, 5, 6. Pentoses, 1, 2, 3, 4, 5 sugars. Okay, so these are deoxyribose and ribose. They are not sugars that we're going to use for energy. These are structural components of DNA, okay, of the nucleotide. These are the 3 that we're concerned with: glucose, fructose, galactose. Okay, that are the monomers of the carbohydrates that we're going to be concerned with. These are parts of the monomer for DNA. And we'll get there. Don't freak on me. Don't freak out. Hold that thought. Okay. One of the things about monosaccharides, they are C6H12O6 no matter what. Hexoses, 6 of them. Hex, 6. So they're glucose, which is sugar for energy, fruits and vegetables. Okay. Fructose, primary sugar in fruit. And galactose is a simple sugar in milk, cheese, and butter and yogurt. Now, they got the same formula, but they all look a little different. So check them out. Okay, it's very obvious that fructose is different because it got a little house-looking thing. Okay, if you look at galactose, they're different right here on the sides. Pretty much the same on all the other ends, but the sides are different. Okay, so just mixing it a little bit, change it from glucose to galactose. So these are isomers having the same formula but different arrangement. This is not isotopes. Iso means similar. Okay, these are mers, meaning arrangement. Okay. Dehydration synthesis, we can put a glucose and a glucose together and we get a maltose and water. Okay, so glucose plus glucose gives us maltose and we get water. Okay. Oh, I don't want to go there. Let's go to the next one. So sucrose is glucose plus fructose. And we get table sugar, sugar cane, or sugar beets. Lactose is glucose plus galactose is milk sugar. And then we already did maltose, which is malt beverages, beer, and malt liquors. Okay. Malted milk balls, the chocolate. Okay. So along that line. All right. Polysaccharides. So polysaccharides are big ones. So more than 2 simple sugars. So go back real quick because these are disaccharides. These are monosaccharides. We do dehydration synthesis to get disaccharides. We keep doing dehydration synthesis to get big ones, macromolecules. So starch are glucose plus more glucose and produced by plants. This is how plants store their energy. Animals, we store energy by glucose plus more glucose is produced by animals. And then cellulose is just a bunch of glucose molecules in a different structure, and they make the cell walls of plants. We term this kind of roughage. Okay. Um, so cellulose, we don't really digest it. That's why cows and other critters can, and really cows chew their cud. Rabbits have got like a little extra pocket, helps them digest it. We don't chew our cud, and we don't have a little extra pocket. Okay. But it does create roughage, fiber in our diet. Okay. If you're thinking of eating celery, you don't like celery, there's no nutritional value in celery. It's all about the cellulose. It's all about the fiber. And if you don't like fungi, here's a reason not to like them, because they have chitin in their cell walls, which again is many molecules of glucose put together. And they're structurally different than the cellulose, but they're in the cell walls of fungi. And that's why a lot of times people can't eat them. Again, get a little bit more fiber in your diet. Okay. So we get into some functional units here, and then we'll finish up carbohydrates. But I'm going to end right here just because I want to keep things like about 30 minutes or so. So I'm going to take a break, going to shut this one down so you guys can digest all that material. Again, we talked about carbohydrates. We talked about monosaccharides, glucose, fructose, galactose. They're isomers, all the same chemical formula, but they differ structurally. We talked about putting them together through dehydration synthesis, and we get maltose. Okay, we get the disaccharides. There's 3 of them: sucrose, lactose, and maltose. And then if we put them together, we can get starch plants, glycogen animals, cellulose cell walls in plants, and chitin cell walls in fungus. Okay, so we're going to end here. Kind of digest that in your brain a little bit. A little digestion, some sugar. And then we're going to pick up and look at some structural stuff. Okay. Thank you again. Get ready for the next one. Digest this little one. If your brain having brain freeze, brain hurting, stop a minute, go back and look at your book and review a little bit. I think I got a worksheet in the Google Drive to kind of take a look at that may help you there. All right. Thank you.